I'm Hana Kiyama, and I'm a student from Makoto Miyata's laboratory and Osaka Metropolitan University, Japan. Today, we'd like to introduce, we are working on the spiral plasma swimming motility by using minimal cell. In the last year's minimal cell workshop, my boss Makoto introduced a reconstruction of spiroplasma swimming in a minimal cell by introduction of spiroplasma 7 swimming genes into Sin3b genome. So by introduction of spiroplasma genes, minimal cell received by Sin3b change their morphology as spherical to helical cell shape, and they started swimming motility. This is a video, and the left side is spiroplasma, and spiroplasma shows helical morphology naturally, and they swim switching left-handed helices and right-handed helices. And middle is original Sin3b, and the right side is Sin3b swimming, which possesses spiroplasma-specific fibril protein and 5 mRNA and hypothetical gene between mRNAs. And they, by introduction of spiroplasma G7 genes, Sin3b changed their morphology to round to helical, and they showed swimming motility like this. To know mechanism and the roles of each proteins, we tried to do the depletion of single protein. But except MRAB5 depletion mutants, there was no obvious effect on Sin3b strains, Sin3b swimming strains. So we try to search for the minimum gene set for the swimming motility. The first, we tried a single protein expression, and MRAB2 and fibril showed a morphological change, but no movement was observed in the single protein expression. So this year, actually we found a minimum gene set for swimming. So we would say this is a minimal motility system in a minimal cell. So that combination was combination of two different mRNAs, and the spiroplasma mRNA can be classified into 3 groups. And these are orange group, yellow, and blue group. So we tried 2 different mRNA combination, so total 10 combination. But at first, we focus on orange and yellow group because there is 2 mRNA in the same group. So we thought this orange and yellow group should be important for the swimming motility. And this is the result. Interestingly, mRNA orange group and yellow group expression, mRNA Sin3b changed their morphology to the helical, and surprisingly they showed swimming motility. And MRB5 and the yellow MRB combination is have more clear helical morphology and swimming motility. So from this result, we can say 2 different MRABs made Sin3b cell helical and motile. In other 6 combinations, no movements was observed. So from this result, we'd like to suggest possible model of spiroplasma swimming evolution. So primitive spiroplasma cells have ancestral forms of the protein, such as mRAB and nucleosides, which will become a fibril protein. And mRAB may be differentiated into at least two groups by probably duplication and accumulation of the mutations. And this step, Spiroplasma acquired fourth generation ability. And these two different mRNAs movements can be transmitted to outside because a spiroplasma molecule doesn't have cell wall. And these post-stimulation movements will be refined into the current motility system, the current swimming machinery, like inside the ribbon structure of the spiroplasma. So our final goal is we would like to see the evolution of the minimal motility in the laboratory from the non-motile cell to the minimal motility by accumulation of the mutation on ancestral mRNA, reconstructed ancestral mRNA. So this time, we tried a kind of opposite direction at the first, from the minimal motility to a non-motile cell by introduction of random mutation by PCR on mRNA5. From this experiment, we can know which residue is important for the swimming. So we screened non-motile cell and the cell which have intermediate motility by the colony morphology. And this is a result. Sin3b typical Sin3b original cell shows a typical mycoplasma colony shape, like fried egg shape. And Sin3b, which have swimming motility by MRB4 and 5, shows a different colony morphology, like distorted edge colony shape, and surface looks like not smooth, a rough surface. And Sin3b, which have MRAB4 and randomly mutated MRAB5, shows both type of colony shape, motile type colony shape and non-motile type colony shape. And half of colony showed non-motile type colony in this time. And we picked up non-motile type colony and various effects on morphology and movements was observed. For example, similar to original Sin3b and the filament cell shape, but no movements. So we tried sequencing and identified mutation points on MRAB5. So far, we found some interesting mutation and morphology or movements like here. So we will continue this screening on a random mutation on MRB5, and hopefully we would like to see the lab evolution of the motility in near future. Thank you for your attention, and the short, simple, slow questions are welcome. Thank you.
Hannah, that is dazzling. I was stunned last year with the addition of just a handful of genes to make a non-motile cell suddenly motile. Once again, Makoto, I am dazzled by the work coming out of your lab. What do spiroplasma colonies look like? Do they have the egg-shaped morphology that you see for mycoides?
To observe this colony shape difference, we used it different condition from normal cell growth. Like this time we used whole serum instead of FBS, and we used the lower concentration of agar, 0.3% agar, and whole serum. So in this condition, spiroplasma colony doesn't look like Sin3b, which have swimming motility. They shows more inside of the agar, and they are wider, wider, spread wider. This.
If we were to use genome transplantation and delete some of the genes that are the Sin3b genes, the mycoplasma mycoides genes, if we were to take out some of the MRAB genes, or the MRAB gene, what do you think the effect might be?
Transplantation of the spiroplasma into my capriculum?
No, what I'm thinking, so you can add genes to Sin3b quite simply, but to take genes away, you have to go through a yeast clone. And so it's a different process to remove genes from the mycoplasma part of the genome. And so I was wondering if we were to remove, if we were to think about removing some of the mycoplasma mycoides genes, how that might affect some of the processes. I don't know if that would be interesting, but it could be done.
Thank you. It's should be interesting. Yeah. There is some. Yeah. Of course. We cannot conclude only MRB4 and 5 induce motility. So there should be important some spacing of inside of the cell, and probably maybe the membrane probably important for the swimming, but I'm still not sure. Thank you for your question.
Can you see what cell division looks like in these cells? Is it like a normal spiroplasma cell division process?
We didn't see the time lapse image of the cell division yet.
Okay. What's next for your, what will you do next?
Yeah, we'd like to reconstruct evolution in the laboratory of the minimal cell, minimal motility. At first we construct ancestral MRIB from the sequence, and we will give them random mutation. This is the final goal of our study.
Okay. Hannah, terrific talk. Congratulations.
John, there's a question in the chat.
Oh, please. Let's see.
I would like to know how you distinguish swimming motility from just random movement.
Can you distinguish swimming motility? We can distinguish swimming motility because reconstructed Sin3b swimming cell shows left-handed switching left-handed helices and right-handed helices like spiroplasma. So we can say this is swimming motility.